Hypersensitivity reactions simply refer to immune responses that harm the body instead of defending it. Normally, the white blood cells or leukocytes and associated mediators which constitute the immune system undergo a series of processes in response to invasion of foreign bodies or pathogens. These series of processes are collectively known as inflammation. In hypersensitivity reactions, the inflammatory process becomes exaggerated, bringing undesirable effects on the body. There are 4 types of hypersensitivity reactions. In this video, we will talk about the first type of hypersensitivity, what is known as type 1 hypersensitivity. Type 1 hypersensitivity or anaphylaxis is mainly identified with the type of antibody or immunoglobulin class that the body produces to attack the invading object. Out of the 5 antibody classes, namely IgA, IgD, IgE, IgG, and IgM, IgE is the antibody which characterizes a type 1 hypersensitivity reaction and so is a good marker for the diagnosis of type 1 hypersensitivity. What causes type 1 hypersensitivity? Type 1 hypersensitivity occurs in response to allergens. Allergens are substances that are generally non-harmful, but the body perceives to be a threat, and so initiates an immune response against. A person who has allergies most likely belongs to a family which has a genetic predisposition that makes their immune system more sensitive to particular substances than other people. Allergens could range from pollen grains, bee stings, strong scented perfumes, animal dander, that is, fur and dead skin periodically shed by some animals, like cats and dogs. It could even be something as common and edible as groundnut or pea. The immune response is quick and usually short-lived. Type 1 hypersensitivity is sometimes therefore referred to as immediate hypersensitivity because of this feature. So how does this reaction happen? Let's take an example. A girl called Abba is allergic to groundnut but has no idea she does because she has never eaten groundnut or groundnut containing food in her life. She tries groundnut soup for the first time, oblivious of what could happen, but her body has a whole different plan. When the groundnut gets ingested, pieces of the groundnut antigen is picked up by antigen-presenting cells, APCs, such as dendritic cells. Dendritic cells are a class of APCs found on surfaces which communicate with the external environment. An example of a dendritic cell is an APC called the Langerhans cell, which is found in the skin. Other APCs include tissue macrophages, Kupffer cells, and B cells, which are concentrated at respective sites in the body. Depending on where the antigen or allergen is found, an antigen-presenting cell picks up the antigen and presents the antigen to a T helper cell using its major histocompatibility complex, MHC. Since this is the first time that the specific antigen in the groundnut has been presented to the T helper cell, it transitions from a naive T helper cell to a primed T helper cell, or Th2, which simply means that the T helper cell has now gained exposure to that antigen. Priming causes the T helper cell to release several inflammatory molecules such as interleukin-4, interleukin-5, and interleukin-10. These are collectively known as cytokines. By default, B cells produce polyvalent IgM antibodies as first-line antibody defense against invading pathogens or substances. The interleukin-4 cytokine, which is produced by the T helper cell, triggers a change in the immunoglobulin or antibody class which is being produced by the B cells in a process known as class switching. Class switching here simply means that the B cells start to produce IgE antibodies instead of the default IgM antibodies. Interleukin-5 from the T helper cell initiates a chemotactic effect on eosinophils, drawing them to the site of the inflammation and causing them to degranulate in an attempt to release inflammatory mediators which may hopefully destroy the invading particle, though nearby host cells may also be affected. The IgE produced by the B cells then bind to the surface of mast cells, a type of granulocytes, via their Fc epsilon receptor in wait for the next attack of the groundnut antigen, if Abba ever eats it again. This whole process describes the first or the sensitization phase of type 1 hypersensitivity reaction. Moving on to the second and more dangerous phase of type 1 hypersensitivity reaction, the reaction phase. Take the analogy of an army private who has been trained to kill enemies in a war, but does not have any knowledge who the enemy is or what they look like. The private is shot in the arm, sustains a flesh wound, but manages to catch a glimpse of the shooter's uniform and looks. From there onwards, he becomes primed to classify anyone who looks anything like his past shooter an enemy and therefore would initiate an attack immediately. Let's say Abba eats groundnut again, several weeks after being sensitized to the groundnut antigen. The sensitized mast cells are now prepared, just like the army private, to fight the groundnut antigen. Upon binding of the antigen to the IgE antibodies already present on the mast cells are signaled to degranulate, releasing pro-inflammatory molecules, which then initiate a cascade of reactions in the body. One of such pro-inflammatory molecules is histamine. Histamine is a vasodilator molecule, which means that it increases vascular lumen diameter and blood vessel permeability. This subsequently increases the amount of blood flowing to the area of the inflammation. Fluids then leak from the vessels to the interstitium and cause swelling. This is what we commonly see as edema accompanying allergic reactions. If the edema persists, surrounding cells tend to absorb some of the fluid, causing them to swell and appear as reddish skin rashes. This describes a condition known as allergic urticaria or hives. Histamine also causes smooth muscle contraction. This phenomenon affects respiration in that bronchi smooth muscles also contract, narrowing down the airway. This phenomenon triggers an asthmatic attack in an asthma predisposed person and could be fatal in some cases. Other chemical mediators produced by the mast cells include interleukin 4, interleukin 5, and interleukin 10, just like the T cells in the sensitization phase. Another set of molecules known as leukotrienes are produced, most especially the B4 and C4 leukotriene subtypes. These chemical mediators attract even more immune cells to the site of the inflammation, including eosinophils, basophils, and more mast cells. Continuous degranulation and release of pro-inflammatory mediators elevates the intensity of the inflammation and sometimes sustains the reaction even when the antigen has far long been destroyed. Under conditions of lower antigenic doses and milder immune responses in type 1 hypersensitivity, symptoms like edema, allergic rhinitis, which is an inflammation of the nasal area, hives or urticaria, eczema, and mild asthma may be seen. However, under conditions of high antigenic doses, the immune reaction could be so severe that a person may be sent into an anaphylactic shock, and this could be life-threatening if emergency measures are not taken. Mild edema could also progress into angioedema, which signifies vascular leakage from even deeper blood vessels. Strong bronchoconstriction could lead to asphyxiation in severe cases. Treatment of type 1 hypersensitivity reaction basically targets the various mediators which cause the unpleasant or life-threatening pathophysiology. Antihistamines are used to reduce levels of histamine and its effects thereof to fight off edema, urticaria, and the bronchoconstriction effects which causes asthma. Corticosteroids or leukotriene inhibitors are also used to reduce levels of leukotrienes and other pro-inflammatory molecules, whilst ingestion of epinephrine causes vasoconstriction, preventing vascular leakage.